We're going to move on to our second speaker in this session, and that's Philip Biggin from the Department of Biochemistry at the University of Oxford, who will be telling us about computational biophysics of trafficking receptors. So over to you, Phil, if you could share your screen, please.
Great. Okay, thanks very much. Thanks for the invitation. Yeah, so I'm going to sort of switch gears slightly and delve into something a bit more atomistic almost. So this is hopefully a small but useful contribution, as I think you'll see, to the computational understanding of cellular trafficking. And the system that we've been focusing on is the KDEL system. So for those, just to introduce this system to you, if you're not familiar with it. So this is the system that's involved in trafficking secretory proteins through the ER, through the Golgi, and then to the plasma membrane. So it's sort of moving from left to right, as it were, on the screen here. And one of the problems is that these secretory proteins, which are indicated in as yellow sort of stars here, as they move through the Golgi and through into the ER, they sometimes drag with them by accident proteins which should reside in the ER, these so-called ER resident proteins, which are these red hexagons. And the cell has to deal with that problem because they get shuffled forwards by accident, and then really we need them to go back to the ER. And the protein that does that is the KDEL receptor, and that's shown here in these as these green sort of blobs, if you like. And they recognize a sequence, or tag, if you like, on the end of these ER resident proteins called, which is basically KDEL, so KDEL. And the KDEL receptor recognizes these proteins, and that initiates the binding of COP1 to form a complex around this, which buds off via a vesicle formation, and then we can begin this process of retrograde transport, where the resident proteins can be released back into the ER, and the whole cycle can begin again. And we don't really understand too much about the molecular details of some of these processes, and we're just trying to understand some of these now. And there are two things to note about this process. One is that as you move from the ER through the Golgi, there's a slight change in lipid composition, which I'm not going to tell you about too much today. But the other thing I am going to refer to, which is important, is the slight pH gradient change. So as you move from the ER, the pH is higher, so sort of 7 point something. And as you go to the Golgi, it's about 6 point something. So it's a very slight pH change, but it's going to be very critical for how the proteins respond and this process proceeds. So our interest in this got sort of piqued by a collaboration with Simon Newstead, who solved the structure recently of the KDEL receptor. This is what this animation is showing you. Hopefully you can see it. Hopefully it's not too jumpy via the internet. He solved this in an APO state and a KDEL bound state. And this is just a morph that's happening on the left, showing you the kind of overall conformational changes that happen. But the one thing that kind of was very interesting for us was this identification here at the bottom of the binding site. So this is the KDEL binding peptide, or the peptide that would be the tag, if you like, of the resident protein. This is just a peptide in the crystal structure here. At the binding pocket, the bottom of the binding pocket, there's an appearance of a very short hydrogen bond between this E127 and this tyrosine 158. And also present or nearby is a histidine, this histidine H12, which we suspect might have something to do with how it senses pH. So there are lots of questions that came about from that initial crystal structure, and these are just some of them. There are actually many more that we're currently trying to address, but these are some of the ones that I'll focus on today. Questions including things like, well, if this really is a short hydrogen bond, is it what's known sometimes as a low barrier hydrogen bond, where the energy to move the proton between them is very between the two oxygens, if you like, in this case, is very low energy, and that has a consequence that it'll be quite a strong, should be expected to be quite a strong bond. So if that is true, then we can ask things about what might the energetic consequences be. Could H12 be a pH sensor? And if that's true, how would that, if anything, would that affect the H bond? And also, does that histidine affect the behavior of the pockets in terms of where the KDEL binds? But there's also a pocket below it, which I didn't show you on that little slide, but I'll come back to that later on. And finally, how does the KDEL peptide actually engage or disengage with that binding pocket? And these are all methods that, or these are all questions that computational methods are quite appropriate for, and can give you some insight that sometimes might appear at first glance a little bit abstract, but I'll hopefully show you how we can pull this all together to improve the overall picture of our understanding of how the KDEL receptor works. And just to add, this is all work that was done by a very talented graduate student called William Wu, and so he deserves all the credit for this work that I'm going to talk about, and I deserve all the flames. So let's just start with the short hydrogen bond. So as I said, this is just a zoom in again of the previous slide, but you can see here the difference between these oxygens in the KDEL bound versus the apostate is 2.5 when the KDEL is bound, but it's 2.8 in the apostate. And that doesn't sound like much, but actually that's quite a crucial difference. And the question we have is that important, and does H12 influence that? But before we get to that, we have to actually try and first confirm where the protons actually might be, because actually we don't actually know that from the crystal data. It's not high enough resolution. So what we'd first of all is try to systematically position all the protons on the oxygens and minimize that with quantum mechanics. So we use quite a high level of quantum mechanics to look at this. And in the apostate, so the idea is basically you put the proton near one of these oxygens and then you minimize it with quantum mechanics and see where it goes. And if you put the proton near the tyrosine oxygen, then it minimizes down and it sits on the tyrosine. If you put it next to one of these oxygens on the glutamic acid, then it goes to where you'd expect it. And that's all as you'd expect. The interesting thing comes perhaps when you look at the KDEL bound state, because if you try to put the proton near the OE1, actually what you see is that it actually minimizes to a position that converges actually if you put it close to the tyrosine oxygen. So you can't actually put it on the OE1, it likes to sit on this tyrosine oxygen. And that was really the first hint that perhaps this bond is actually one of these so-called low barrier hydrogen bonds. So I wanted to look at that in a little bit more detail. So we did a few more detailed quantum mechanic calculations in this instance. And this is a little bit of a complicated slide, but the idea here is that we look at the energy that's required to move the proton from being bound at the tyrosine, okay? So that's this position here. And then what we do is we plot the energy as we move the proton, transferring it to basically one of these oxygens on the E127. And we do that in the presence of the histidine in different protonation states. So if it's got a proton on the delta or the epsilon nitrogen, so just to remind you what that is, just showing you the histidine here, there, and it's neutral, then this is the blue and orange lines. And then we can have a protonated histidine where both nitrogens have hydrogens present, if you like. And you can see for the apostate, the graph looks, there's a steep barrier, there's a well here, and then there's a steep barrier, and then you enter another well down here as well. Okay, so, and this barrier here is pretty high, so 15 to 20 kcals, and that's pretty consistent with previous reports of this kind of process. Now, the interesting thing comes perhaps when you look at the KDEL bound state. So very different profiles now. You can see here, starting, there's a little well where the tyrosine, when it's on the tyrosine, and then you can see here actually that it flattens out, and particularly so for when we've got the protonated histidine. So you can see here, this is actually very flat. This dashed line here is what's called the 0 point energy, which refers to, in this case, the energy of the OH stretch, the vibration of that OH bond anyway. So you can see here that the flattening here means that it's very easy for this proton to want to move, if it was on this glutamic acid, to move down into the well. So that's entirely consistent, of course, with what I told you in the previous slide about trying to minimize it on OE1. So that tells you two things. One is that it looks like H12 has some influence. Even in the apo state, there is some influence of H12, even though this barrier means that it's not going to be actually meaningful. But certainly there's some influence here. And it also tells us that in the KDEL bound state, the protonated histidine would allow this transfer of a proton potentially, and that it's a very low energy barrier to move the proton between those two oxygens. Now we can do something a little bit more advanced. So that was all quantum mechanics, just quantum mechanics. The next level of treatment is to think about the whole system. So in those previous calculations, I just showed you they were based on a very sort of cut down system, just to examine that in the level of QM. But we can also do a QM-MM calculation, and we can now try and compute the free energy that it's required to move between the two states, depending on where you start from. So this is a little bit maybe hard to interpret at first glance, but if you start from the apo state, so remember the hydrogen bond between those two atoms, between the Y158 and E127, will be over here somewhere. And then the idea is that we try and squash that, okay, trying to stretch it to move them to shorter distances. And you can see here, if we try to do that, it doesn't really matter whether we've got histidine protonated or not. You can see that the energy rises pretty steeply as we try to push that closer together. If, on the other hand, we start from the KDEL bound state, okay, and try to stretch the bond towards what it would be in the apo state, then you can see here that there's a well, obviously, for KDEL bound here. And as we try to stretch that, then you can see we move to increasing energy. And the important thing here is that the protonated histidine also would favor you staying in the KDEL bound state. So there's an energy penalty for moving away from that KDEL bound state that's increased when the proton is histidine. Again, this is all kind of consistent from what we learned in the previous two sets of calculations. So apo doesn't look like it can form this short hydrogen bond, and in the KDEL bound state, where you can form this short hydrogen bond, and indeed that is an energy well, even in that state you need the histidine, the protonated histidine will actually mean that you would need more energy to perform the separation if you wanted to do that. So that was kind of interesting from looking at it from the perspective about whether the histidine influenced the short hydrogen bond, but we can actually ask the question the other way around and say, does the short hydrogen bond influence the H12 protonation state? And to do that, we do molecular mechanics, but free energy calculation. I'm not going to bore you with the details. And we do that with a kind of efficient way, in a rather efficient way. We do two calculations at the same time. And that's what I'm showing you how this is set up in reality. We basically have an apo state here and a KDEL bound state here. And we do two simultaneous free energy calculations in the same box, if you like. So we calculate the deprotonation free energy of H12 in the KDEL bound state, and simultaneously we calculate the protonation of H12 in the apo state. And so what you end up with is a delta G. That may be hard to get your heads a little bit around, so I'll just tell you what it means, these numbers mean. What this means is that this number here of 3.55 and so on here in this first row, these are just different independent runs of the calculation. You can see that when we have the short hydrogen bond imposed, that there's quite a significant energy. This is about 3.5 kcals. So it's energetically unfavorable to deprotonate the H12 in the KDEL bound state. We have to kind of cheat a little bit here because the molecular mechanics is not designed to reproduce a short hydrogen bond. It doesn't really know how to do that. And so you can see that by if we just sort of let the restraint go, then the number drops a bit, and that's reflective of the system actually adapting during the calculation to where it likes to sit in the energy minimum of the molecular mechanics force field that we're using. But just to give you an idea of and these other numbers here are just random histidines, just to give you an idea of the change that you might randomly expect to see, basically. So you can see it's basically 0.5 or 0.9 of a kcal for a random histidine somewhere else on the protein. So this this is the amount of energy that would be, it's unfavorable to deprotonate the H12 by when you're in that KDEL bound state. So that's nice, but that tells us something about the energetics, but where does that energy come from? And if you remember, I told you, or I showed you a movie of some of the conformational changes that happen, and mostly focused on the H7, which is that red helix moving outwards. But actually, if you look inside the protein, there are also some quite significant changes. And in the apo state, these gray and orange blobs are kind of the volume of the pockets that exist inside the receptor. And you can see here there are two pockets, a gray one and an orange one, either side of the central hydrogen bond, which I've been telling you about. And in the KDEL bound state, this gray pocket shrinks quite a lot, and so does this orange guy as well. And there's also the appearance of a small red one as well. So the KDEL binding reduces cavities both sides of the hydrogen bond that's formed, and that's quite important. So, oops, just go back a bit. So the cavity volumes in the apo state are such that actually there's no difference in the water networks that form in there. So if you look at this is a neutral pocket and looking at the water density or the pattern of water density that we see in the pockets, and you can see that that's very similar to the situation when we've got histidine protonated. So the water networks are essentially the same. On the other hand, if you look at the KDEL bound state, then actually there's a very important difference. So in the neutral state, you can see there's a few, maybe 2 or 3 waters here that are sort of sitting in this sort of configuration here. But in the protonated state, then you get quite a lot of chaining of about 2 or 3 more waters that like to sit here. And this is probably where all this extra energy comes from. The protonated histidine 12 allows you to form most of these additional hydrogen bonded waters here. Okay, so what does all that mean for the receptor biology in the cell? These are quite abstract calculations to some extent, but how can we relate that back to what happens in the cell? That's after all what we're trying to really understand here. And so we can come up with this kind of schematic model that summarizes this. So in the top row, so this top section here, we're in the Golgi, okay? So we're at slightly lower pH. And so the idea is that this histidine, this is H12, will be protonated, okay? Then bind KDEL. This enables a short hydrogen bond to form, and this is a very stable complex. Or we don't know whether this is dependent on that or vice versa, but this is the situation you end up with. You then have this recognized by the COP1 complex, and then this will then be trafficked into the ER. So all of this in the bottom row is the ER, okay? And in the ER, the higher pH means that this histidine is likely to now be neutral, okay? And that will then, of course, then move the distribution of water pockets back to what we saw in the apo state, which will help the KDEL to dissociate. And we think we then go around full circle, and you can then begin to then complete the process again. So that's kind of how we think the receptor works. And just to sort of add one last thing to this, which we're sort of beginning to also look at, is to try and compute or look at how KDEL binding is actually or proceeds and is recognized. And we can use something called steered molecular dynamics to do this, to try and calculate a free energy of the process of binding. So basically, as the peptide moves down into or out of the binding pocket, can we compute the free energy for that? And that there's a movie showing you hopefully on the right of what's happening, but the energy is shown in this dashed line here. So we're moving towards the binding pocket. So this would be bound down the bottom here. And what's interesting is that there's a kind of handshake or a ladder of arginines. So you can see here, this is the hydrogen bond occupancy of these arginines as we move down into the configuration, into the bound configuration. So you can see R169 acts first, and then R5, and then R47. And then finally, you end up with this, with the K of the KDEL talking to this E117. And that's actually another whole lot of questions about the role of E117, which I'm not going to have time for, but hopefully we're still working on that as we go forward. So I'll just summarize what I hopefully have told you. And that's that the short hydrogen bond impacts on the ability of H12 to protonate and vice versa. And that also has a controlling effect on the behavior of the pockets. And that finally, I hope I just showed you that just very briefly that this arginine ladder looks quite interesting in terms of helping how the peptide engages or disengages with the receptor itself. And it's very complex. This whole thing is, I hope I've given you some idea of the complexity of how everything is interlinked in this process. And there are many questions which you might have and which I might not know the answer to that might arise from this work even now. And that's something that's still ongoing. So just end by just thanking all the people that are in my lab and helped with this, but particularly Will, this is Will again, who did all the work, and also Simon and Francis Barr in Oxford, who this is a big collaboration with. And I'll stop there and take any questions.
Thanks, Philip. That was really good. Chris, do we have questions on the Q&A?
Yeah, we do. So since the ER and Golgi membranes have different membrane properties, have you considered the effects of lipid environments such as packing, curvature, and their electrostatics?
Yeah, that's a very good question. And that is something that we are currently pursuing in quite a lot of detail. So we're looking maybe not so much at curvature perhaps, but we are definitely looking at things like cholesterol, saturation, the amount of saturation in the lipid species there and that sort of thing. And we're hoping to, that's some ongoing work, but it's going quite well. I'm quite confident we can say something interesting about the differences in the membrane compositions that might have an effect on the way that the receptor works.
Yeah, okay. Another question is, have you thought, or what kind of experimental approaches might you apply that could support some of your computational findings?
Yep, that's a good question as well. So yeah, all of these things we obviously try to go back and test with our colleagues or try to suggest some experiments. So we can do things like it's very hard actually, before I get to that, it's very hard actually to be sure the short hydrogen bond is real. So you noticed I framed it very carefully in the sense that if it is real, then this is the consequences of that. Beyond that, we can do things which I didn't mention there, but we can do things like suggest mutants, for example, in the recognition process, which can then be confirmed, and we can ask, and Simon can do an assay for that. We can also do things, or Francis's group can also do things where they try to do tagging in cells and live cells and try to see whether what we're saying is true or not. And there are various ways to do that. That's something that we'll hopefully see more of in the future. That's only just really started to get going.
Okay, and we have a question from Andres, who's on the panel. If you could unmute yourself.
Thank you. Thanks for the very nice talk, Phil. So well, my bias is for QM and QMMM, but I was wondering if you tried ProPKA or H++ and how they perform.
Yeah, so you can do ProPKA and things like that, do an okay job, but it's not that accurate. So if you really want to understand the finer level of accuracy, it's quite difficult to, well, ProPKA, first of all, won't necessarily do a very good job of that, and you have to be a bit wary. It's fine for giving you a rough idea, but beyond that, it's not that good. Having said that, actually doing even QMMM calculations of protonation states, absolute protonation states, is actually still quite tricky in as far as I know, and getting the absolute values right. Relative values is okay, and so if you're working in a relative framework, then you can do okay with that, but an absolute calculation is still, I still think there's some problems with that.
I think we can take one more question.
One more, okay. Another, well, a very common question, I'm sure, that you come up against, particularly in view of the participation of water in your result and your observations. So the model that you use for water, how sensitive is the result to that effect?
Yeah, that's a very good question, and although we haven't done it particularly in this work, in other work in the lab, we have looked very systematically at the waters and different force fields in general. And in fact, usually we do try to do studies where we do at least 2 force fields. And in this case, you're right, we probably should do more water models as well. We have done other studies with water models, for example, in permeation of ion channels. In our work with that, we have looked very specifically at different models of water, and that there are some effects. I think a lot more work will be needed to really disentangle all that, including polarizability, for example.
Okay. A very quick question from Maria. Yeah, from Maria.
Sure. I think you muted, Maria.
Yeah, you muted Maria.
Sorry. Thank you, Phil. This was a really nice talk. It was a bit about the experimental methods. Are there mutants of this receptor that you could use to validate? Because if the histidine seems to be really important to direct everything, would you mutate a histidine and see if there is still binding, or what is the behavior?
Yeah, there's been lots of mutagenesis done, which and Simon is still doing that. There's lots of in some cases quite surprising mutagenesis that you think would affect things, but actually it doesn't. So I can tell you probably that the E117, which if you remember is at the top of the binding pocket, and it looks like it's critical for binding the lysine. But actually, you can mutate that, and the thing still binds pretty well, no problem. So there's an interesting set of that's what I was referring to actually. And there's a lot of other interesting questions to do with the binding, which we're still working on, and hopefully we can probably present something on that at another time. But there is a lot of mutagenesis done in-house, but there's also a lot of things that are known about different species having different tags, and Francis has done a lot of work on the the distribution of those different tags in the cell as well. So all of that is known, but not publicly known.
Okay, thank you.
Thanks a lot, Maria, for your question. Thanks very much, Phil.